Biologists have been using plastic labware for decades. Plastic is tougher than glass and breaks less easily, is lighter, and is less expensive. And plastics are disposable. Use a plastic tube or microplate once, then throw it away. But most commercial plastics have one serious drawback, namely leachables. As their name implies, leachables are chemical compounds which, through normal use, seep out of plastic vessels and into your experiment. Leachables are mostly small molecules and metals that arise or are introduced during the manufacture of polymeric materials. The leftovers of processing typical laboratory plastics such as polypropylene, polyethylene, and polystyrene. The main sources of leachables in laboratory-grade plastics are plasticizers, slip agents, metals, solubilizers, and antioxidants. Plasticizers reduce brittleness in polymers. Slip agents make plastics easier to remove from molds, and antioxidants confer long shelf life on plastic products by slowing polymer breakdown. Metals may be used to catalyze polymerization. Since these materials are not incorporated into the polymeric chains but persist in the final product, they can leach from the surface of tubes, plates, or pipette tips. Certain solvents, high temperatures, and buffers can exacerbate leaching of these agents from plastic labware. As long ago as 1980, researchers noted that neurite growth slowed or stopped after 4 days of culturing in plastic dishes. Since then, we've learned that leachables can bind to proteins or DNA, thereby interfering with observed interactions among proteins, genes, drugs, and reagents. Leachables can inhibit or inactivate enzymes, disrupt the activity of proteins, and even interfere with spectroscopic analysis. And as we've seen, leachables can affect the normal growth of test cells and disrupt normal cellular function. In short, leachables introduce inconsistency, uncertainty, and variability into your experiments. You've probably heard about the reproducibility crisis in the life sciences. Why take chances with yet another variable to control? The key to minimizing the potential impact of leachables is to keep tight control over every step in the production of plastic labware, from the polymerization of raw materials to final production to manufacture and function testing. Some companies, like Eppendorf, ensure their disposable labware is produced from virgin polypropylene, polyethylene, polycarbonate, polystyrene, or UV transparent plastic. These companies never use recycled plastics, nor do they employ slip agents, biocides, methyl dodecylammonium salts, plasticizers, melamine, silicone, or latex. It is also important to only use materials from suppliers who guarantee that animal-derived components, when they are used, are sourced from countries that have not been affected with bovine spongiform encephalopathy. No plastic can ever be entirely free of leachables, but using laboratory consumables from quality sources greatly reduce risks associated with plastic labware. Using laboratory consumables from companies like Eppendorf lets you work in confidence and spend more time thinking of your experiment and less time worrying about leachables.